Okay, good morning or afternoon, everybody, whenever you are watching this video. I apologize about a little bit of personal history. At the moment, unfortunately, I'm suffering from a very bad attack of gout. Gout is a very extremely painful condition. I have it in my foot. This is relevant to what we're talking about today. This is not just to get your sympathy. Unfortunately, I have to take some medications which may sometimes make me extremely ill, especially in the morning, which is what happened this morning. But let's talk about the gout. Gout is a condition of the joints, and it is extremely painful. Well, as soon as I say that word painful, you understand what I'm talking about. You know the sensation of pain. This is interesting. How do we know a sensation of pain? Second thing, I have gout in my big toe. It's as far away from my brain as you could possibly get. And yet I know perfectly well, because it's extremely painful, that I have pain in my toe. How do I know I've got pain in my toe? Well, first of all, there are specialized nerves all over our body which perceive pain. When those nerves fire off, when they send a signal, our brain knows it is receiving information from a pain receptor, and it interprets that sensation as pain. In actual fact, the message it receives is no different to the message it receives from nerves in your eye that get interpreted as vision. It's the same thing arrives in your brain. It's just that your brain knows it is arriving from a pain receptor. That's one thing. The second thing is even more marvelous. How does the brain know that it's in my big toe? How? I mean, we automatically say, oh, well, the pain's in my big toe. Is it? Is the pain really in my big toe, or is the pain up here? The pain, the construction of the whole scenario is here in my brain. But my brain has a map. It has a map of the body. And it can place a sensation. And what the brain does is it projects that sensation. It projects the sensation to my toe. So I feel it in my toe, but the feeling is actually up here. It's actually in my brain. We know that this is true because in some cases, the brain has a poor map of the area where something is going wrong. And often when that is the case, the pain will be felt inappropriately. It'll be felt somewhere. You may have something happening in your gut, but you actually have a feeling in your shoulder. That is called referred pain. And it's because the brain is having a hard time mapping where the sensation has come from. Anything from our hands, our feet are very well mapped indeed. The reason why should we have pain? It seems like such an inconvenient thing. We can actually not only feel pain, but we feel different kinds of pain and different degrees of pain. And it's actually extremely useful and very valuable in terms of survival, because it means you'll protect the wounded or injured or malfunctioning part of the body. You'll protect it, not overwork it, and allow healing processes to take place, not aggravate the situation. So all of these things that we simply accept, oh, I've got a pain in my toe, are actually extremely complicated. And they reflect on this amazing system of communication. We've discussed one major system of communication already, and that was the endocrine system. That is a system of communication. Remember, we kept on talking about how different parts of the body talk to the hypothalamus, for example, and then the hypothalamus talks back. It's a system of communication, but also of coordination. That is the one, the endocrine system is the one great system of communication and coordination. But the nervous system is the other. The nervous system is the other way in which the body coordinates all of its activities and directs all of its activities. So that's what we're going to talk about today. Central to the functioning of the nervous system, of course, are these specialized, very specialized cells, which are nerve cells. And the nerve cells are distinguished because they are able to generate two kinds of signal. And the first is basically an electrical signal. And that electrical signal can travel for a very long distance. It can travel for meters. My big toe is hurting. I'm perceiving it up here. My brain is projecting down to it. In other words, messages are going back and forth and back and forth and back and forth over meters of neuron. The proper term for the cell is the neuron. Nerves are something a little bit different, which I'll distinguish in a minute. We often talk about nerve cells, though. It's the same as the neuron. So it's the primary way system for nerve cells is they can generate an electrical signal. But the second is, and something which you might not immediately realize, is that nerve cells are also secretory cells. Indeed, some nerve cells we have heard about, like those of the hypothalamus, that could themselves secrete endocrine substances, hormones. But the nerve cells, all nerve cells have the capability of producing special chemical signaling molecules. The electrical signals travel over a long distance. The purpose of the chemical signals is to travel over a very short distance, as we'll see in a while, and they're essential to the nerve's functioning. So we're not going to talk much, well, at least in this class. In the next class, I'll talk a little bit about the structure of the brain and everything. But it's really important to bear in mind that nerve cells don't just exist on their own. Neurons actually interact with one another in very, very complex fashions. There are two parts of an organism where extensive interactions actually take place. And the first are in small clusterings of nerve endings which have their own little linking nerves called interneurons. And these form small bodies called the ganglia. And especially in our autonomic nervous system, that's the non-voluntary nervous system, extensive processing may take place in the ganglia. But the major integration of nerve information, of neuronal information, takes place in a much larger collection of neurons and interneurons, which we refer to as the brain. The brain is really a feature of bilaterally symmetrical organisms. And in the chordates, the brain is formed from a neural tube. Even our brain is actually our brain and spinal cord are an elaboration of a tube structure with a hollow center. So these, whether it's ganglia or the brain, what is happening is that nerve impulses are coming in as sensory input of some sort. They are being integrated in one way or another. In the brain, they may be extensively shunted around different areas of the brain for interpretation. And then finally, an output is made. That we refer to as the motor output. The response of ganglion or brain is to send out information, send out instructions, and that is a motor output. Whether it actually results in movement or not is immaterial. It's still referred to as a motor output. It may, in fact, have other results other than muscle movement. So, first of all, we've already talked about the fact that there are sensory neurons all over the body which are picking up information. Those sensory neurons are extremely diverse, and they pick up all sorts of information which is relayed to the brain. It may be relayed to conscious parts of the brain, but most of the information is actually going to unconscious processing of the brain. And when we look at our nervous systems, what we'll see, we have obviously the brain is very, in our case, really large, and a spinal cord, and that is referred to as the central nervous system. This is the area where that integration and processing of information takes place. Then we have an extensive nervous system outside of the central nervous system called the peripheral nervous system. And that is all of the nerves which carrying, sending information to the brain and then carrying away the motor instructions from the brain and from the ganglia as well. So at this point, let me just draw this distinction between neuron or nerve cell and nerve, because we incorrectly tend to use those interchangeably. In actual fact, a neuron or nerve cell is an individual cell with its particular structure. A nerve is a bundle of neurons, and it's usually surrounded by a protective connective tissue sheath. So have a look here. These are spinal nerves. This is a spinal cord here, which has been dissected. These are the vertebrae here. And you can see the root there of the spinal nerves. But have a look here, and this is a nerve there. It's a bundle of neurons, all surrounded by a connective tissue sheath. And by the way, you see these lumps here. These are ganglia. There's specialized ganglia associated with the spinal cord. So these are nerve roots, and then they all gather together. These axons, we'll hear later. And they bundle together, and this is, we call a nerve. This is a large, visible collection of neurons. Okay, so let's have a look at the structure of the nerve cell itself. It's essential that we understand this, because it's central to the whole functioning of the nerve, as we'll hear. So first of all, let me point out that this is a nerve cell. We can think of it. This is the nerve cell in my big toe yelling at me to tell me that my toe is hurting. So you can think of it as the terminal neuron. It is a sensory neuron. It's picking up some sensation. And this is the structure of the nerve cell. It has a cell body there. And the cell, it's just like every other cell, is nucleate. And we'll point out at the start, nerve cells are extremely metabolically active, very, very active for two reasons. The first is that the generation of the nerve impulse that we'll talk about in a while takes a lot of energy. It involves all sorts of protein pumps that are actively moving ions back and forth. So that's the first need for energy, is running all of these pumps. The second reason that it uses a lot of energy is that nerve cells are secretory. And what they are secreting are largely neurotransmitters. And we'll see that in a minute, exactly how that works and why they are there. So when you look at a nerve cell, you actually see it has inside the cytoplasm, it's going to have a lot of endoplasmic reticulum. It has a very active Golgi body, et cetera, et cetera. They're very, very active cells. They also have a high metabolic demand, and they have a high metabolic demand for oxygen because they respire quite rapidly. So that's the cell body. Remember that what we're looking at is a sensory nerve, but this is typical for many nerves. The cell body is extended out into fine extensions called the dendrites. And the cell membrane of the dendrites is often highly modified so that it will respond to particular stimulus. Okay, either if this is pressure, then this would be modified, that this will actually generate a nerve impulse when it's pressed. If it's a heat receptor, same thing, it will respond to heat, but your heat receptor is not going to respond to you pressing on it. That's the idea. These can be very specialized neurons. So these are the dendrites. They are receiving sensory information of some sort. When a nerve impulse is generated by this nerve, actually, I'll just point out to you right now, you see this area here, this kind of funnel-shaped area leading to this extension here. This is called the axon hillock. And that is important because this is where the nerve impulse is actually generated. It's right in this path, this belt of cell membrane. That's where the nerve impulse is initially going to be generated. So that this long extension of the cell here is called the axon. And it may be short, but it may run for meters. Some axons are very, very long, like those that form the spinal nerve that I just showed you. They may run all the way from a limb up to the spine. So the idea is that this cell is going to generate a nerve impulse of some sort. It's going to generate an electrical impulse. And that electrical impulse is going to travel all the way down here. It's going to travel only in this direction for reasons that we'll hear. But ultimately, this axon has to end. It has to end somewhere. In this case, it's communicating with another cell. But when you look at the end of that axon, you look at all of these little ends here, they call the axon terminals or the synaptic terminals. When you look at them, they don't stick close to the cell membrane of the succeeding cell here. And the electrical impulse, which is generated in this axon, cannot jump this gap. This is a very good insulator in actual fact. It won't transmit an electrical impulse. So the electrical impulse ends here. Well, this is a difficulty. How is this cell going to communicate then with this cell here? The answer is, we'll discuss it again in a little bit more detail later on, but the answer is, remember, the nerve cell is also a secretory cell, and it is secreting substances called neurotransmitters. And when the nerve impulse, the electrical impulse reaches this here, it induces the cell to release its neurotransmitters. And the neurotransmitters diffuse across this gap. It's very small. They diffuse across this gap, and they are picked up by this cell here. This is the synapse here. That gap is called the synapse. And the postsynaptic cell is this one here. It picks up those neurotransmitters, and the neurotransmitters themselves cause something to happen inside the nerve cell. In this case, we'll just say they cause this nerve cell to generate a new nerve impulse, and off it goes down its axon to whatever its destination is going to be. So those are the basic structures that you need to remember. There are a couple of elaborations that we'll talk about in a while. And the cell body, dendrites, axon hillock, please remember it, the axon hillock, and then the axon terminal or synaptic terminals. This is the postsynaptic cell that it's communicating with, and it has its own axons, etc., etc., etc. Okay, so when you look at the nervous system as well, especially in brain, spinal cord, and in large collections of neurons, when they're put together in these functional bodies, there are a lot of associated cells which hang around. And these are referred to as the glial cells. They're very, very complex. And their importance is the subject of a tremendous amount of research, because they had formerly just been thought of as being kind of support cells. There are many kinds of glial cells, and they have different kinds of origins. Some of them are actually modified macrophages, blood cells, white blood cells. Others are derived from other sources. And they are usually thought of as being kind of support cells. But it appears, research suggests that they are tremendously important in maintaining, if you like, a kind of hygiene of the brain, especially in the spinal cord, in that they clean up proteins, extraneous proteins, dead cells, this sort of thing. But they also seem to play a very important role in the structuring of memory. It's not exactly understood how, but the glial cells are these kind of support cells. They're not nerve cells, but they're associated with the nerves, and they have many different complex functions. These, by the way, these are glial cells here, but these are neurons here. This is actually in the cerebellum of the brain. Here's the cell body here, and these are very, very complex dendrites which interact with one another. Here, it's a very complex structure. Okay, so now we need to talk about the nerve impulse. You've probably heard of that term before, nerve impulse, but how it arises is actually quite a complicated thing. The first thing I would explain to you is, I've told you before that a cell membrane is a boundary. It's a barrier. Even though it's just these two lipid molecules thick, those lipid molecules form a very powerful insulator. Electrical charge, ions don't cross them very easily, and electrical charge can't get across. It's an insulator, an electrical insulator. You've heard me say this before. If I have an insulator and I have lots and lots of negative charge on one side, masses of electrons on one side, and I have very few on the other side, that's positive on the other side, and I separate the two of them by an insulator, what have I got? I've got a battery. If I connect the two sides somehow with a wire or whatever, electrons will flow from one side to the other, and I can get those electrons to do some work for me. This is really analogous to what we're going to look at in the nerve cell, because in the nerve cell, the cell membrane acts as a barrier. And if we come back here, what we're going to find is that the electrical charges outside here are different to the electrical charges in here. There is a net positive charge on one side and a net negative charge on the other side. So we've got positive charge on one side, negative on the other side, on either side of this narrow barrier. What we've done is, when that occurs, we have created a system of tremendous amount of potential energy. The cell can actually harvest that potential energy and use it. In this case, it's going to use it to generate a moving signal. And that is the nerve impulse. So we're going to need to understand exactly how this does. And the first thing to tell you is that if we look at the concentration of ions around nerves, we'll find that it's rather strange, because outside here, there are a tremendous amount of sodium ions. They're Na+, okay, it's a positive charge. And there are also some potassium ions, K+. There are far fewer, in fact, there are rather few sodium ions on the inside of the cell. There's potassium, but not much sodium. There's far more sodium outside than there is potassium inside. there are far more positive charges on the outside of the cell than there are inside the cell. And the inside of the cell is therefore relatively negative. In addition, all of the huge molecules like the DNA and the proteins and everything else, all of those giant biomolecules tend to be negatively charged. And that adds to the fact that the cytoplasm in here is relatively negative compared to the outside of the cell, which has lots of positive sodium ions and is relatively positive. We can measure that. We can measure that difference, and we'll hear about it, exactly what it is in a minute, because it's an important figure. So here's one of the reasons why the nerve cells are so metabolically active. And that is they have very active pumps called the sodium-potassium pumps. Sodium-potassium pumps are a complex protein in the cell membrane, which continuously pump sodium out to maintain that high concentration of sodium ions outside the cell. And they pump potassium into the cytoplasm of the cell. So we end up with this powerful gradient across the cell membrane. It's a twofold gradient. First of all, there's a gradient of charge. It's more positive outside the cell than it is inside. But also we have these very powerful chemical gradients. There's more sodium outside trying to diffuse into the cell. There's more potassium inside the cell trying to diffuse out. This creates a situation where there is a lot of potential energy across that cell membrane. And that's we're going to follow to see what happens. So let's just here's the basic scenario. We have sodium-potassium pumps in the cell membrane, which are continuously pumping sodium out and pumping potassium in. There's more sodium outside than there is potassium inside. And so there is a relatively negative charge across this membrane on the cytoplasmic side. In addition, remember, we have these other biomolecules which are negatively charged, which add to this negative charge. The cell works very, very hard to keep, to try to maintain this gradient. At the same time, though, potassium is allowed to leak out through potassium channels. And so there are also sodium channels, but there are very few sodium channels. There are more potassium channels. And some potassium does leak out to try and achieve equilibrium with the very few potassium ions that are on the outside here. Every time a potassium leaves here, that's one less positive charge. When you allow the potassium, some of the potassium to diffuse across like this, it increases the negative charge here. But notice that the cell works really hard to pump to pump sodium out here. In fact, the sodium-potassium pump, when it works, it moves three ions, sodium ions, that way. For every 2 potassium ions, it moves that way. That's one of the reasons that's one of the ways in which this differential is managed. So we can come now and we can actually measure this difference here across, and it's about -35 millivolts across. So now we need to pay careful attention, because this we need to get clear in our heads before we proceed to try and understand how a nerve impulse is actually generated. We need to introduce you to another set of channels, of ion channels. You realize now ion channels are tremendously important. We've already talked about. Here's an ion pump. And here are passive channels that allow diffusion, potassium that way, and sodium this way. But these are passive channels. We need to talk about some active channels. And there are 3 that are particularly important. And these are gated channels. A gated channel is one which can be closed and opened under different circumstances. And there are three types. The first is a ligand-gated channel. A ligand-gated channel is one which can bind a substance and open. So it will only open when it binds a particular substance. Well, that would be typical of, let me go back all the way back here. That would be typical of the situation here. It's not the only place that occurs, but that would be typical of this situation here. We would expect to have ligand-gated channels here, ready to bind this neurotransmitter and open to allow a nerve impulse to be generated. So that would be a ligand-gated channel. There are also sensation-gated channels, like the pain receptors, heat receptors, etc., etc. When those nerve cells are stimulated by their particular sensation that they're specialized for, they have channels which open to generate a nerve impulse. But the ones we're going to spend most of our time talking about are the voltage-gated channels. Voltage-gated channels respond to a very specific voltage. Remember that a voltage is a measure of potential electrical energy. And voltage-gated channels respond to a very particular voltage. When that voltage is reached, they either open, in some cases they shut at a particular voltage. There are different ways in which voltage-gated channels function. So remember that. These voltage-gated channels that we're going to see, they are going to respond to the presence of a differential, of a potential, of an electrical potential. So we also need to understand some terms. When we look at the membrane of a neuron, we'll see that normally on one side it is positive, and then on the inside of the neuron it is more negative. If I swap that over, if I decrease the amount of positivity on the one side, so I'm decreasing the potential across, that is referred to as a depolarization. But I can actually also have situations where I could increase the differential, and that is referred to as a hyperpolarization. So in the nerve cell, I told you there's lots of sodium outside and there's not so much sodium inside. If I allow sodium to flow in to the cell, I'm decreasing the potential. I'm depolarizing by allowing the sodium to flow into the cytoplasm. That we're going to hear a lot about. But I can also hyperpolarize. I can actually pump potassium out, for example. I can pump potassium out of the cytoplasm. And by doing that, I would increase the negativity of the cytoplasm. That is called hyperpolarization. And we're going to see a good example of where hyperpolarization also occurs. Depolarization is what's going to catch our attention first, because it is depolarization that creates the nerve impulse that we're going to talk about. So here's another thing about these gated channels. That is that the number of gated channels that open are going to be proportional to the stimulus. So if I just stimulate a nerve a little bit, a couple of channels will open, bink, and then they'll shut, bink, then they'll shut. But there's a critical point. If I stimulate strongly enough, I'm going to see that all of my channels will fly open all at once. We refer to the nerve impulse as being an all or nothing response. And I'll try to explain it a little bit just by showing you the structure. If we're sitting at a particular point on a nerve, right, and we watch what happens right there at the point where we're standing on the nerve, We watch the changes in voltage as a nerve impulse passes. This is what we'll see. Remember that this picture here is for one particular place on the nerve, on the cell membrane. So we're going to, first of all, nothing's happening, right? The nerve cell is just sitting quiet. There's no stimulation. There's no nerve impulse passing, nothing like that. Let's just say that we're right at the axon hillock, because this is the critical place where a nerve impulse is going to be generated. Okay, so a couple of the dendrites of the cell get stimulated one way or another. Just a couple. When that happens, we're going to see that there's a slight depolarization. First of all, let's notice the cell is sitting here, it's resting. And that means it's got a relatively negative inside and a relatively positive outside. And the difference between the two is about minus 70 millivolts. Here's the scale here, minus 50 is here. It's about minus 70 millivolts here. That is the resting potential. If I just give a little bit of stimulation, it'll depolarize, it'll become more positive. It'll become more positive. But let's just say it's just a little bit, it only goes here, only comes up here. I can stimulate several dendrites at once and I can get a bigger response. This is called a graded response. But the graded response simply dies out. It doesn't, nothing happens until we reach a threshold. If I stimulate enough and I depolarize enough, I reach a threshold here. It's actually minus 55 millivolts. And at minus 55 millivolts, something spectacular happens. And that is that all of the voltage-gated sodium channels suddenly open in this immediate geographic area. Now we are on the axon hillock, right? Right there in that region where all of that stimulation is being received from the dendrites, all of the voltage-gated sodium channels open and sodium floods into the interior of the cell. Well, what is going to happen if suddenly I equalize the charges? I'm going to depolarize. I'm going to make the inside, I'm going to depolarize. It's going to become much more positive. In fact, it goes to 35 millivolts. And at 35 millivolts, the sodium channels suddenly slam shut and potassium channels open. And potassium now begins to flood into the cell. And as a result, the cell repolarizes. In fact, it goes a little bit too far. It hyperpolarizes there. There's much more potassium inside the cell now than we actually is required. And now those sodium potassium pumps that we talked about earlier, they start frantically working to reestablish the resting potential. So what you can just think about it this way, the really important voltage-gated channels to understand are these sodium voltage-gated channels, all sitting in the cell membrane. But if I stimulate a particular part of that cell membrane strongly enough, if I can stimulate it so that we depolarize to the threshold, as we reach minus 55, in that immediate geographic area, all of the sodium voltage-gated channels fly open and the sodium floods into the cell. The cell depolarizes, but when it reaches here, it flips, it slams shut the voltage-gated channels, sodium channels, and opens potassium channels to allow the cell to repolarize. So this is hyperpolar, and it does it a little bit too much. You usually get a little dip here. That's the hyperpolarization before the sodium potassium pumps return everything to the resting potential. We'll do it several times. We'll do it in sequence, okay, and follow along. Here's our normal situation. Here's the cell membrane of the neuron, and here are voltage-gated sodium channels here. Okay, here's a potassium channel. Potassium, this is an ordinary potassium channel. It's closed. This is the normal resting situation. The sodium-potassium pumps, which aren't shown here, are pumping sodium out, and they're pumping potassium in. And they do so until we reach this this minus 70 millivolt resting potential. When we stimulate an area of the, we'll think about the axon hillock area. So we're receiving stimulation from the dendrites arriving at the axon hillock. And every time a little bit of stimulation arises, one or two of those sodium channels may open. But it's very little. And it just dies, it simply dies away. But they are additive. These stimuli are additive. So if enough of them, if enough of the dendrites are stimulated, for example, or if they're stimulated repeatedly or very strongly, then the stimulation, this depolarization that results from opening these channels reaches a threshold, and that threshold is minus 55 millivolts. And when that happens, when minus 55 arrives, suddenly all of the sodium channels open. They all fly open in that geographic area, and sodium floods in. And as a result, the potential of the membrane flips. We depolarize. Look what's happened. Look here, we've got positive on the outside, negative here, positive, negative. When it depolarizes, the charges on the membrane flips. All of a sudden, we've got masses of sodium, positive sodiums on the inside and fewer on the outside. This is now relatively negative here, relatively positive there. And this is reflected here. If we were measuring the voltage, we'd measure up to 35 millivolts up there. That's the peak of the action potential. As that happens, as it reaches 35, what happens is that all of these sodium channels slam shut. And now the potassium channels open. And as a result, we begin to repolarize the inside of the nerve cell. And this is proceeds until such time as we have re-achieved the you can see positive on the outside, negative on the inside. It overshoots a little bit there, but the sodium-potassium pumps soon restore the resting potential there. So what we're watching is that this changeover of the orientation of charge. We normally sit positive on the outside, negative on the inside. But as this action potential is generated in a particular area of the membrane, we have a flip. We have a depolarization that leads to positive on the inside, negative on the outside, and that soon is restored to the normal situation. So that tells us what is happening immediately in that particular area where we're standing, right? We're seeing this sudden, what if amounts effectively to a flow of electrons, a flow of charge rather, from one side of the membrane to the other. It's the only thing that's a little bit difficult to understand is the fact that we're talking about two positive ions, okay? But you need to remember that we're talking about net charge, okay? And under normal circumstances, there are far more positive charges on the outside than there are on the inside. There are far more positive sodiums on the outside than there are positive potassiums on the inside of the cell. Relative charge is more positive outside than it is on the inside at resting. As the action potential is generated, that flips, it depolarizes. There are some important things to note, and that is that this is an all or nothing response. When we reach this threshold and we generate the nerve impulse, it generates a nerve impulse, always generates a nerve impulse, 35 millivolts. That's it. It's set. It is set by the responses of those gated channels, of the gated ion channels, at minus 55 and at 35. Okay, that's when the channels respond, and they either open or they slam shut. It's an all or nothing response. But remember what I told you. This is what we would see if we're standing at the 1 point. So how is it then that this gets to travel down the nerve? How is it that this signal, this electrical signal, doesn't just happen at one place and stop? Well, I must explain, by the way, before I tell you about this, because it's an important thing. In this period here, when these sodium channels slam shut again, they cannot reopen there's a period of time, seconds, when they cannot reopen. This is called a refractory period. It's important for a reason you'll see in a second. So I said seconds, probably milliseconds, I'm sorry. And this is called a refractory period. And even if you stimulate them really hard in the refractory period, they won't open at all. So let's just say, I started off by saying actually our nerve impulse is generated at the axon hillock. And that is because the axon hillock sums up all of the information that is feeding into the cell body through dendrites and through other nerves that are impacting on the cell body as well. That is, if the axon hillock is depolarized, then an action potential is generated, which will travel down the axon. But why does it travel down the axon? Why does it travel in one direction only? Well, the answer is actually simple, and it's complicated. The first thing is that we said that we depolarize in a geographic area, right? We don't instantaneously depolarize the whole membrane. That would be chaotic. We want our impulse, our nerve impulse, to be controlled and to travel in a set direction. So the opening of those channels occurs only of the sodium channels. The depolarization only occurs in a set area. So here it is here, okay? Here's the place. Let's say this is our axon hillock. And we depolarize, we allow all of those sodiums to flood into the cell here. Well, they actually don't just stay like in a blob. They actually diffuse. And if sufficient of them diffuse, then they will depolarize this part of the membrane here. And if sufficient of them diffuse to depolarize enough of the adjacent membrane, then those sodium channels in this area will now open. So you can think of the ions themselves, sodium ions themselves, as leaking to the side. And when they leak to the side, they stimulate another action potential adjacent to them. Here's why that refractory period is important. Because in actual fact, they could be stimulated. Look at this one here. Okay, let's say we've stimulated another action potential here. This one could actually stimulate these ones to open, but they can't. All of these sodium channels are in their refractory period. They cannot open. The only ones that can open are on this side. The only channels that can open are on this side. When this generates the next one here, the only ones that can open are these ones on this side here. And as a result, this is the direction that the nerve impulse, the action potential, will travel. It will only travel in that direction down the axon because of this brief refractory period behind it. It stops the action potential from going backwards. Meanwhile, behind, of course, we are reestablishing the voltages across the membrane, as we saw before. Okay, so that's fine. That we've got our nerve impulse traveling down the axon. It does it quite fast, but in actual fact, it does take time. It can take fractions of a second to travel a meter or so, but that is measurable time. It is not particularly, and in terms of the nervous system, the action potential sometimes travels relatively slowly in some parts of the nervous system compared to other parts. And what I mean by that is, in one section of our nervous system, which is the autonomic nervous system, nerve impulses travel relatively slowly compared to other parts of our body, particularly those which are under voluntary or motor control. And there's an important reason why this is so. It's because skeletal, the nerves, for example, which feed skeletal muscle, all the voluntary muscles, etc., have a special structure. And that is that the axon is actually insulated. The axon has a lipid wrapping around it, which acts as an insulator. This is produced by specialized cells called the Schwann cells. Here's the Schwann cell here. And if you cut through a neuron, what you will see is here's a Schwann cell here, and it's wrapped around in the spiral around the axon of the nerve. And this provides a very powerful electrical insulator. And as a result, where the nerve is covered there by the Schwann cell, the Schwann cell has this lipid in it called myelin. And wherever the axon is covered by a myelin sheath, it does not allow depolarization across the cell membrane. And in fact, underneath these Schwann cell areas here, you can see them here. Underneath those, there are no sodium-potassium channels underneath there at all. Instead, all of the channels are clustered at the gaps in between these insulating layers. So this here is a Schwann cell. It's forming this myelin sheath around the nerve, and the gaps here are called the nodes of Ranvier. So, or the nodes of Ranvier, however you want to pronounce it, it's fine. So these are the nodes of Ranvier, nodes of Ranvier, and all of the sodium channels are clustered here. And this leads to a very strange situation, and that is that they are close enough together that they can respond to depolarization, but they are separated from one another, so that the action potential actually jumps. It actually jumps between these gaps. It's called saltation. Instead of generating an action potential at each and every area of the cell membrane coming down here, instead we only generate an action potential there, but that is powerful enough to open these channels here. And the action potential appears to jump from node to node to node to node. This speeds up the passage of the action potential prodigiously. And the nerve impulse travels much faster down myelinated nerve sheath than it does down non-myelinated. There's actually, by the way, there's another way to increase the speed of the passage of the action potential, and that's to increase the diameter of the axon. That's not important for us because where we need fast conduction, we myelinate the axons. But in other animals, like for example in the squid, they don't have myelin. They don't have the myelin sheath. And where they need to have fast conducting nerves, they increase the diameter of the nerve. They actually produce a cell called a giant neuron, which has a huge, thick axon to conduct nerve impulses really fast. So remember this is the process of saltatory conduction or saltation, the jumping of the action potential from node to node. All right, so now we need to talk a little bit about how nerves communicate with one another, because this is central to the processes of integration. A single nerve cell can receive information from another nerve cell. And in fact, a single nerve cell can receive information from many nerve cells. Not only that, a single nerve cell can receive different kinds of information from other nerve cells. So it provides a possibility that nerve cells can begin to link with one another in very, very complex ways in order to analyze, integrate, and produce a sophisticated response. That is exactly what our nerve systems do in our brains. Not only that, nerve cells can also communicate not only with other nerves, but they can communicate with gland systems. They can communicate with organs. They can communicate with muscles. They can communicate with many different parts of the body. And in order to do so, they have to be able to take that all or nothing action potential and convert it into some sort of useful signal. And the whole problem is that the axon ends, and it ends at the axon terminal. And somehow or other, the information has to leave the neuron in such a form that it can be communicated to another type of cell. And that place where the axons end, where the axons terminate, is called the synapse. And the communication across that synapse, we've already heard, is by means of a neurotransmitter. And we'll see some of the functions of that in a minute. First of all, the neurons are secretory cells, and they continually secrete. They do not respond to a nerve impulse by secreting, by making neurotransmitter. Instead, they normally produce neurotransmitter in abundance, and they store it. They store it right there at the axon terminal, and they release that neurotransmitter in response to the arrival of that all or nothing action potential. That neurotransmitter then has to diffuse across the gap that comprises the synaptic cleft. It has to diffuse from the nerve cell itself to its destination, whatever its destination may be. So we'll have a look here, and you'll see that here's an axon terminal here, and stored at the axon terminal in vesicles is the neurotransmitter of whatever kind that neurotransmitter is. In this case, this cell is communicating to another nerve cell. And these here are ligand-gated ion channels. Now, there are many kinds of ligand-gated channels. they can produce different effects. But for the moment, we're just going to think about this one here. When the ligand, when the neurotransmitter binds here to this channel, it opens. And what happens is it allows potassium to flow out and sodium to flow in. Well, think about that. What are we talking about now? We're saying we're allowing potassium to flow out, and we're allowing sodium to flow in. We are depolarizing. We're depolarizing there. And if enough neurotransmitter comes across, if enough, the amount of neurotransmitter is proportional to the number of action potentials that are arriving, by the way. If enough diffuses across here, then we can generate a new action potential at this place here on the nerve cell. So that is the way in which the neurotransmitter is going to have an effect on the succeeding cell. But it all depends on the identity of this nerve cell here and the identity of the destination cell. In this case here, they have used a postsynaptic neuron. So the neurotransmitter here is causing the generation of another nerve impulse. But this could easily be a gland cell being stimulated to produce an endocrine secretion. It could be a muscle cell being stimulated to contract, something like that. All right, so let's leave it there for today, and then I'll continue this and talk a little bit about nerve systems in our next class.